are shorter today. I hope we'll have a very short presentation, and then we can start our revision series. We'll wait for a few minutes, so then to allow people to join, and we can start. So we have 2 weeks left before the end of this class. So I think I'm meeting some of you today. I'll be in my office until 3:34 for the ones who haven't emailed me and who want to meet. Just send me an email so that I'm aware. And I will be holding office hours next week and the week after. And the other week, the 12th, I think, no, the 13th before the final exam. So I will be in my office to meet one-on-one. I may email some of you because I said that I will, the one to whom I said I was going to email before the class, the exam. So I'll email you. But I'll be more than happy to meet on one-on-one in the coming weeks. Just let me know. We can have an online meeting anyway, but I will have so in person. So there will be this week, next week, the week after, and Tuesday 13th. Yeah, just 2 days before the 2 or 3 days before the 3 days. Let's wait for a few minutes and then we can start, so then I won't have to repeat. Oh, it has already started. I'm making sure it's recording today. So it is recording. Last week it didn't record, so I just recorded maybe 10, 15 minutes maximum. I'm going to post this. I haven't posted this yet to the ones who missed it. Okay, so good morning, everyone. It's a recording, and I will to start. So just before starting, kind of a housekeeping. You might have seen this. So today we'll have a brief or general overview on plants. I was planning to have more in-depth, to give you more in-depth information about what are the chemical reactions that happen, when, for example, when we cook potatoes and when like we do and other. But then I had to cut my slides to give you an overview and to save time to start our revision. So I have already prepared a few slides for revision. I'm going to do this for the coming 2 weeks. Next week, same. We'll have our lecture on food chemistry and innovative technologies. Same, we'll have second part, we'll go for revision, and then we'll have a tutorial. For the tutorial, Sylvia will be here and will be helping to facilitate this. What we didn't do in sensory analysis, if you remember, in sensory analysis, I asked if there were volunteers to participate, because it was about tasting food so I was not sure if all of you were comfortable with this or not. So this is why I asked for volunteers, but maybe it would have been more interesting to have all of you involved. So this is what we'll be doing the next week. So everyone should be involved. You guys will be working in small groups. So you'll be given one food, and then based on the theory that Sylvia will be presenting at the beginning, so you will be trying to identify the methods that can be used to analyze food. So this is what we'll have. Last week of class, last week I didn't have the survey as I said, because so I said, okay, to save time, this is what we will do. I will record the lecture slides before the lecture. So it will be overall about how we do fortification. So like very brief introduction about food fortifications, vitamins, etc. So you will have the slides recorded. You will have the slides, PDF, and the transcript for the ones who want, who are interested. So then we will dedicate almost 2 hours of the lecture to the revision. So this will be our revision session. And then the last, around 11 a.m., we will have the tutorial guest lecture. And if you remember, I told you this last week. So I invited a PhD candidate who is working in the same lab as Akash, who was working in a food fortification. So, and I thought it would be more interesting that you guys can see and have an idea about what is done within the Department of Nutritional Science. So these are our, so this is for today. And if you remember, I told you last week, we will have, so this is what I do, for what I have been doing since COVID started. So I usually organize an online revision session before the exam. So we'll do this for the ones who are interested. So I will implement a survey to see what's your availabilities. And as I said, the online revisions won't be recorded. So if you cannot make it, then we can meet. I'm happy to meet on one-on-one. So this is this is an overview about what we'll have for the coming 2 weeks. And then, as I said before starting, you're more than welcome to come and to ask for a one-on-one in-person meeting or online. So I'll be more than happy to do this. So today's lecture will be, let me put my timer. So today's lecture will be on plants, mostly vegetables and, sorry, let me just put my timer. Okay, so we'll be talking about vegetables and fruits. I'll be giving you mainly an overview about chemicals and what is the importance of these chemicals in fruits and vegetables.
And I will be giving, so first section, second part will be giving you examples and what can be done, or what are the main chemical reactions, as I said, or main examples. So let's start. So we all know that fruits and vegetables are of significance. So based on dietary guidelines for Canadian, so there are over half of adults, about 59, now they consume less than recommended daily intake. So they consume less than 5 to 7 fruits and vegetables per day, which is a concern. And in this, I wanted to share with you a paper that was published in Lancet about 3 years ago. For people who don't know, Lancet is a very high impact factor journal, so like very prestigious one. And they look at an overall intake on fruits and vegetables over the world to see what is the gap between consumptions and the need of fruits and vegetables. So for the one who are interested, you can go and read this paper. It's a very interesting one. I think it's an open access, so you don't have to access it through UFT. Why it's very important? So we will look at today at the importance in fruits and vegetables in food science or food processing. So why they're important? Because of high or sugar content. So they can be used as they are, or we can use their compounds, such as sugars, pigments, etc., for food processing. I will give you some examples. For example, for the starch, the starch can be consumed as it is within the foods, within the food matrix, or within the fruits or a vegetable, or can also be extracted to be used for food processing. Same, for example, for the chlorophylls or the carotenoids. So these are compounds that are emerging to be used as food additives during the processing because they are considered as natural food additives. So they can be easily added, and there is no concern about adding chemicals in food during the food processing. So these are naturally extracted. So when we talk about chemical composition of fruits and vegetables, these are highly dependent on the photosynthesis and absorption of water and minerals during the growth and the maturation on the parent plant. So everything depends on the growth of this fruit and vegetables. So we'll see there are reactions that happen before harvesting, and then there are reactions that happen even after harvesting. So it depends on fruit. We will see the climacteric and non-climacteric later in a few slides. But then overall, after the harvest, so after when we collect the fruit or the vegetables, so the source of nutrients are no longer available because the fruits and vegetables, they get their source of nutrients from the soil. So all the minerals from the soils, they absorb chemicals. For example, they can even absorb pesticide, etc. And they get water. There are chemical reactions that I'm not going over here, etc. So at the end, here there is no source of nutrients. And then these sources of nutrients, or macronutrients, or even micronutrients, they are considered as what we call independent units. So sugar content is a sugar content, so it doesn't change unless there are maturations. So, and then these are considered as independent units. So what is there? It's there, in which the respiratory processes play a role in this. So if there is a respiration, if a fruit is respirating, so these chemical reactions or these independent units may react together to have or to produce, for example, ethanol, to reproduce gases, etc. So here, when we talk about respiration, we'll usually talk about uptake of oxygen and loss of CO2 and H2O. Overall, fruit and vegetables, they have high content of water. It ranges between 70 to 90% of the fruit or the vegetable itself. So they have higher water content. This water content is usually associated with high water activity, but it depends on the fruit and the vegetable. So it has a higher content of carbohydrate. They have higher content, relatively higher content of carbohydrate, but they have very low compared to other products. They have very low content of lipids and protein. We'll see later. So the major constituent is the water. So this is why fruits and vegetables can be highly contaminated and can be highly susceptible to microbial growth, etc. So this is why. To avoid this, so there are different ways of doing it. We either dry the food, fruits or vegetable, mainly dry the fruits. Fermentation can also play a role in increasing shelf life, and different canning, etc., and different processing. So all to control. So in the plant cell wall, so as I said, there is a water content. So in the plant cell wall, water is as a part of a matrix gel. So these vegetables or fruits, so they act as a matrix or within a matrix. And then here, water can play a role as a wetting agent, so to make the product wet or to preserve high water content, as a solvent for minerals, for micronutrients, solvent for minerals and vitamins, as a structural component, because it is a part of this cellular composition or the food or the fruit matrix, fruit or vegetable matrix, and then as a polymer stabilizer. So the water acts as a stabilizer as well. So because it helps making this matrix, and then the matrix may help stabilizing the other components within the gel, or the matrix gel, as we call it here. Here, just to give you an idea about the carbohydrates that are present. So so the reference is missing, but it's one of the links I will tell you later. For the one, it's a modified from a book chapter on vegetables and fruits and vegetables. So here, this is just to give you an idea about sugar or carbohydrate content. So, for example, you can see the main sugars that are present. We have glucose, we have a fructose, we have a sucrose, and in less extent, the maltose. So, as you can see here, you may have noticed that these are different colors, and I did it in purpose. So, glucose and fructose, these are non-reducing sugar, right? They're non-reducing sugars. And sucrose is a reducing sugar, right? I'm correct, right? Go. I said the opposite. I am testing you. So, reducing sugars, glucose and fructose are reducing sugars because they have free hemiacetyl to precipitate. The sucrose, if you remember, I said, oh, this is a not reducing sugar, and we have, so there is, to make it a reducing sugar, we have, it can be precipitated, and it can be subject to an enzymatic reaction and an enzyme called invertase. You need to remember this. Okay, so here, and the maltose is in different maltose, for example, I bold it in grape. So because maltose is one of the substrates that are uptaken by yeast and bacteria for grape fermentation. So for example, making wine, etc. So the maltose is the main substrate for the lactic, so mainly lactic bacteria, and or what we call them malolactic bacteria. This is just for your knowledge. Malolactic bacteria, because they use the maltose to produce an acid. And then here I bolded broccoli and onions. The broccoli and onions, they have, in addition to the main sugars, they have two other sugars, raffinose and the stachyose. Raffinose and the stachyose, we call them RFOs. So what are these RFOs? These are sugars. They belong to the raffinose family oligosaccharides. They are mainly present in fruits and vegetables. And why do we call them like this? So we'll see. So raffinose, so when we talk about RFOs, we talk about mainly two sugars, but there are other ones. But the main ones, the raffinose and the stachyose that are present in the vegetables, fruits and the vegetables I told you about. So we have raffinose oligosaccharide and we have stachyose oligosaccharide. Stachyose is one of the most abundant in pulse and legumes. In the legumes, it's one of the most abundant sugar or polysaccharide, and it's called alpha galactosides. They're called alpha galactosides because these are they can be galactose and glucose, but they combine it together with the alpha linkages. So this is why we call them alpha galactosides. So what do we know about the raffinose? Raffinose is a tree sugar, so it's three saccharides. So it is, as you can see here, forget about the last one. You just look at the tree. So it is a galactose plus a glucose plus a fructose. Glucose and fructose, if you remember, in combination together, they make sucrose. This is from the first carbohydrate lecture, part 1. So together they make, so when we talk about the raffinose, we talk about the galactose that is combined with the sucrose. But at the end, these are three sugars, and they are linked by alpha-1,6 bonds. So this is, so they're linked with alpha-6 bond. And then obviously, if you remember, so glucose and the fructose, they have beta-1,2 linkages. Why it's my thing is weird. Do you see it like this in your PDF?
No, it's normal.
Huh, this is weird. Okay, so this is 3-sugar raffinose. Then we have 4 sugars. They are stachyose. So as I said, it's one of the most abundant in legumes. It's a tetrasaccharide because they have 4 saccharides in total. So it's a sucrose plus 2 galactose and with alpha-1,6 linkages. Or it is a raffinose with 1 galactose. So this is what we call the stachyose. The RFOs can be, they have 2 opposite effects or 2 opposite functions. They can be considered as anti-nutrients. You will see why. And they can also be used as beneficial or beneficial sugars or belonging to fiber group. So, so why anti-nutritional can be used as an anti-nutritional factor? Because it causes flatulence in humans and in animals. Why? Because in humans or animals, there is a lack of enzymes that are called alpha-galactosidase. So what these alpha-galactosidase come? So they come and they cut the galactose, which make them more digestible. So if we don't have these alpha-galactosidase, this sugar, so these two sugars or arafos, they are not digested in the small intestine. As they are, 3 tetrasaccharide or 4 saccharide, or 3 saccharide, sorry, they reach the colon, non-digested because there is no alpha-galactosidase. When they reach the colon, they are fermented by microbes in the colon. And it's called anaerobic fermentation. Hi, Akash. It's called anaerobic fermentation because it happens without oxygen. When we have an anaerobic fermentation, so we have a production of gases, CO2 and other gases. So this is what makes the flatulence happen. In the meantime, these arafos, they are considered as fibers. So they can be uptaken or used by beneficial microbes such as lactobacilli and bifidobacteria. And their consumptions or their fermentation, put aside the anaerobic fermentation that we don't like, and another way, so it's like this happened in the meantime a bit, by the way. So bacteria, they have enzymes, the alpha-galactosidase, which they can digest and consume this. And studies have shown that consumptions of these legumes, for example, containing the arafos, may increase the amount or the level of beneficial microbes in the gut and decrease the level of non-beneficial or pathogenic microbes. So the good will be up and the bad will be low. So this is why they have opposite sides, these arafos, once digested. And then this is why they're considered as potential prebiotics because of their beneficial effect that is mediated by the changes in the microbial composition. So I put this here. What do you think? Why did I put the exclamation and interrogation mark here? Which enzyme? I just said it maybe 2-3 minutes before. I said it. I heard something here. No? Sorry? No. Yeah. Yeah, I just said it. I said it maybe 2-3 minutes. So invertase, because this is a sucrose. So an invertase, this is from carbohydrate lecture, fructose and so invertase here, and then the sucrose, like these arafos as they are, they need to be digested. So monomers and dimers of saccharides or other components can easily be digested within the small intestine. So but these ones, as they are larger molecules, they reach the colon and digested. Protein, we will wait for Akash. I am not saying anything. Akash, wait for you. No pressure. But I'm happy to see you here. So then for the lipids. So if you remember, I said the lipids, they are not, maybe I shouldn't be saying that, but in fruit and vegetables, in terms of processing, importance during the processing, etc., they're not as important, for example, as water, or as important as carbohydrates. Here, the lipid content in of fresh fruits and vegetables is small. So it's about 0.1 to 1%. It can go maybe 2 or 3. You can see in the preference. So, but it is significant in terms of storage and quality of the fruit and vegetable itself. But they rarely contribute to the food quality, etc., especially during the food processing. So they can lead to undesirable flavor changes resulting from the oxidation. So this is in, remember, so because of this lipid content, although it is low, but they can be oxidated. Unsaturated fat within the fruit and vegetables can be unsaturated and may lead to oxidation or oxidation within the fruit, either in the fruits or vegetables. So the lipid of fruits and vegetables, with the exception of olives and avocado, because these are have high content of lipids and lipids, so they are largely confined to the cytoplasmic layers, in which they are especially associated with the surface membrane. So membranes, they have high fat content. So the fat, if for example we see that the lipids are about 0.1. So the 0.1 is mainly present within the surface membrane as a part of the membrane of cellular envelope, for example. So these are for the lipid. I'm not going to go further on the lipid. So the focus was on the carbohydrates and on other minor compositions. So within the minor composition, minor compounds that are present in fruits and vegetables, we can talk about organic acids. So when we talk about organic acids, we have two major examples. We talk about aliphatic acids. These are the ones responsible for the acidity, that is, acidity of fruits and vegetables. So we talk about mainly citric acid, malic acid, tartric, and oxalic acid. So here I'm giving you examples of acid composition. These are in milligram per 100 gram of fruits or vegetables. So we have the malic and the citric acid that are the main or the major one. And we have others, such as tartric, the oxalic, etc. And I put in red the oxalic acid for the rhubarb. So because the oxalic acid, they're known to make this intense acidity in fruit and vegetable. And this is where comes for the ones who have tasted or like tried the rhubarb either as a jam or in a pie, etc. They are very intense. So the acidity is different from lemon and from others. So this acidity comes from the oxalic acid, and as you can see, it's in a higher content. Tartric acid in the grape, so the tartric acid can be associated, so can help in, it is not involved, so when we talk about fermentation, we talk about sugars, etc., but this will help increasing the level of, or differentiating like the taste, or increasing the taste of acidity for fermented grapes. So it comes from the tartric acid that is already present in the grape before the fermentation. Fermentation, as I said a few slides before, fermentation of grape, it's mainly due to production of malic acids. malic acid by lactic bacteria. Maybe. And then we talk about aromatic acids. Aromatic acids because they have, so phenolic acids because they have these cyclic, these are cyclic forms or structures. These are mainly involved in the color and the taste, not on the acidity, and the taste of fruits and vegetables. These are, when we talk about enzymatic reaction, when we talk about Maillard reaction or either other enzymatic reaction, these aromatic or phenolic acids are the substrates of the decoloration or browning or why did I say Maillard reaction? No, it's non-enzymatic reaction. So, and then they, for example, when we talk about browning of potato or apples, these are the ones involved. Here, that's just to give you an idea. So I skipped this. So when we talk about aromatic acids, one of the main known and present in fruits and vegetables, we talk about the chlorogenic acids. So chlorogenic acids are ester of caffeic acid and the chenic acid. So like the combination, if you see, they're combined together. But then in the analysis, when we are analyzing fruits and vegetables, we mainly talk about caffeic acids. So caffeic acids, you can see they're here. They have some fruits and vegetables, they have higher content of caffeic acids. So, and then this caffeic acid, there are studies showing its beneficial effect on different beneficial effects. And then one of the beneficial effects, for example, of these fruits and vegetables can be due to the presence or high presence of caffeic acid. So this is just to give you an idea about these phenolic acids. When we talk about minor ones, we also talk about phenolic compounds, polyphenols mainly. So what are these polyphenols? They are mainly secondary metabolites of plants. They are, for the plants, these polyphenols, they are needed for the defense against UV radiations or aggression by pathogens. They can even be microbial pathogen or even microbial pathogen, or it can even be other predators. So these are the main, for fruits and the vegetables, this is the main function of polyphenols. So when we talk about polyphenols, we talk about can be divided or distinguished by their structure. So we talk about monomer. Monomer is like one molecule. And we talk about polymers, and here we talk about tannins. So within the monomer, we have phenolic acids. Yes, phenolic acid, they are acids, but they are considered as polyphenol because of their structure. And for example, when we talk about polymers, we talk about the condensed tannin, the proanthocyanidins. The proanthocyanidins, which we call them by abbreviation PAC, are the ones present in grape. They are present in cranberries. Cranberries are very highly known high source of PAC or proanthocyanidin, and we have other ones they are present. So when we talk about polyphenols, we have we can distinguish between their function or their benefit they use as in food, their interest as a food flavoring compound, but they're also interested in health as in health benefits. So when we talk about these polyphenols, they can be source of bitterness. And when we talk about bitterness here, it's different from the one we spoke about last week about when I told you about bitterness of cheese. So when you talk about bitterness of cheese, it's of flavor. That is something that you don't need to have in the cheese because the taste is like very strong and you don't like it. But then here, the bitterness can be something interesting. It's like it brings an extra taste or like a different taste. So and then here, the bitterness, for example, when we talk about grapes, we talk about wine, we talk about even chocolate. They may have some polyphenols that can be present or added from the cacao, etc., that are responsible for this taste. When we talk about bitterness, we have in human there is one that is more studied receptor. It's a taste2R, and this is the one responsible for this sensation in mouth when we eat, for example, berries, or even when we take cranberry, for example, cranberry juice, etc. So these are sensations. They're due to bitterness, and it's because of presence of receptor. In astringency is like it's another sensation of it's like a mixture of acidity and all together. Astringency, we talk about, so the one responsible are the polyphenol. We talk about polyphenol in tea, in grapes, in wine, and other components. So here just give you another. Receptors of astringency can perceive or receive both the bitterness and astringency, and so far there are about 25 receptors that have been identified in humans. The mechanism is not completely understood, although different receptors have been identified, but the complete mechanism has not been understood. And then when we talk about health benefits, so remember I said large molecules are not digested in the small intestine. So for example, this pin of polyphenol, once ingested, I'm talking about larger molecules, or even monomers and dimers. So these polyphenol, once ingested, they reach the intestinal small. The small intestine. So they reach the small intestine. The monomers and dimers, because they can easily like, translocate and go. Here I think you see it on your slide. So this is the blood circulation. So they go to the blood circulation, they reach the liver, and this is where they're metabolized. We're talking about monomers and dimers. They can easily be metabolized by phase 1 and phase 2 enzymes. The polymers, this is why they're interesting. The polymers, they are not digested. So see here, like there are two arrows, one continuous and one discontinuous. The continuous, because they translocate, they go to the liver, they're metabolized. The metabolites of these monomers and dimers can come back to the intestine. The polymers, they reach the colon and digested. And they are uptaken by microbes. These microbes, they have specific enzymes to cleave at specific, they have alpha, for example, alpha glucoside, etc. They cleave, and they, we cannot talk about fermentation. Here it's just a metabolism. They catabolize the polyphenol into different molecules. So then these metabolites, they can go to the systemic circulation and to reach specific organ. And this is why we're interested in polyphenol, because these polyphenol, they can act as a monomer, if they are a monomer or dimer. But then the polymers, they can involve microbial intervention, they can involve different mechanisms, and this is why there is an interest on this. Okay. As I said, the tea is a source of polyphenol. It's a source of can be a source of a good source of antioxidant because they have antioxidant activities. And when we talk about tea, we have to distinguish between green tea and black tea. So the green tea has a catechin. Catechin is a polyphenol. And then the black tea has two other polyphenols. So these are, remember we are here. We're at this polyphenol. We're talking about tannin. So the tannin, they are high molecular weight. So polymer, so they're high molecular weight. So as I said, they're responsible for astringency. So what is the astringency? Is the drying and pickering of the whole surface of the mouth and the tongue. So it's like a mixture of acidity and very, very unique sensation. So tannin, why they're as important? Why do we feel this sensation? So astringency can come from like astringent or astringent, and this means protein precipitation. So, and here we know that the tannin, they bind to the pronilish protein in the tongue. So here, as you can see here, so these are the proteins, and they can bind, and then here that are circulating in red, these are the polymer or the tannin. So and then, is it 45? Oh no. So these are tannin. So then here, the tannin protein complex that is responsible for the astringency. And here I just give you, it's like so that you can distinguish between the phenolic, hydroxyl, etc. And so this combination that makes us feel the astringency of tea and other groups or other fruits. Here, these are extra slides. It's a very interesting paper published this summer about bitter and astringent substances in green tea. And as you can see here, it's they have a spider plot. So look, it's very interesting. One of the very few recent papers that are giving the spider plot. So the paper, for people, this is an extra slide. For people who are interested, you can go and read this review. Same for here. I gave you the link. It's another review talking about possible mechanisms of astringency. As I said, if you remember, so the mechanism of astringency at bitter is not completely understood, although there are some different mini receptors that have been identified. This is another interesting review published recently. Talking about minor composition, we talk about pigmentations. These molecules that are responsible for coloring food. So we talk about the carotenoids, we talk about chlorophyll, and we talk about other ones. So here we can distinguish four. The carotenoids, the ones that are a group of yellow, orange, red, fat-soluble pigment. They are fat-soluble one. They're present in the lipid material of the cell, and they're along with the chlorophyll. So they can be co-present with the chlorophyll. And then here I'm giving you examples of groups. So when we talk about carotenoid, we can distinguish, we can have two subgroups. We talk about the carotene and xanthophyll. And within this, we can talk about the alpha carotene, beta carotene, lycopene, etc. I'm giving you, we'll give you a few examples later. Chlorophyll, the one responsible for the green color. So they are found in oxygen photosynthetic organisms. They can be, they're here in the plant, but they can be in other organisms that use the photosynthesis. Remember I said these components are also interesting in another way. There is emerging evidence showing that chlorophyll can easily be used as food additives to replace the addition of chemicals in food. Because of their coloring, mainly because of their coloring function, they can also be used for other functions, but they're mainly for color. And then we talk about the anthocyanidins. Anthocyanidins are water pigment phenolic groups. These are the ones responsible for the color red, purple, etc. in fruits and vegetables. Anthocyanin, they have been associated with different health benefits, anti-cancer, they alleviate or they mediate, they help with cardiovascular disease, etc. So these are the anthocyanin, and the same for the anthoxanthines. So the anthoxanthine, we have the flavones and the flavonols. So these are water-soluble. So carotene, for example, are fat-soluble. These are water-soluble pigments which range in colors from white to colorless and creamy or to yellow. So, for example, the cauliflower. So this color is due to the presence of anthoxanthine, and the same for the anthocyanin, they have been associated with different health benefits. This is another interesting review for people who are interested in reading more about the anthocyanins. So these are they're mainly known for antioxidant effect, and there are studies showing their effects against cancer, etc. For people who are interested. So moving to another one, then the second part. So we'll be talking about fruits. When we talk about fruits, we can distinguish between palm fruit, stone, berry, citrus, chard, and even tropical. So these fruits, when we talk about fruits, banana or others, like first thing we think about in food chemistry, we talk about browning or enzymatic reactions. So we have seen this, browning of apples, etc. So for example, why apple turns brown? We said it, we have been talking about this since the beginning of class. Because polyphenol, we keep saying it's because of an enzyme called polyphenol oxidase, Yes, it is a generic term. So of a group of enzymes that catalyze the oxidation of phenolic compounds to ketones. These are polyphenol oxidase. They are a family of oxidase of enzymes. So we don't have one, but we have multiple ones. It depends on the substrate. I'll give you an example in the next slide. In this case, the oxygen is required for the reaction to happen, because otherwise there is no browning reaction. And the ketones are highly reactive. So they're electrophilic compounds. They form melanin, which is a brown color. So the melanin formed here, this is the one that's responsible for the coloring. Melanin binds proteins, and here we have enzyme and substrate in the separate cellular components. So they mix together when the tissue is damaged. When we cut the fruits, so this is why we're damaging the tissue, and then we are putting the enzyme and the substrate in contact to each other. So intact, for example, an open apple doesn't brown, doesn't change of color. But once we open, the opening helps in putting them together. And here, this is like kind of a complex and more complex overview on the reaction. We have a phenolic compound, as I said. Okay. We will add a few minutes. We started doing this like 45 minutes, break 45 minutes. And we're extending. Okay, let's make an exception today. Then we are done, and we do. So when we talk about, so a substrate is a phenolic acid, a phenolic compound. So here, this is tyrosine, and it's an amino acid that reacts with an enzyme that is called the tyrosine. You have it here. I just put it in red on my slide, but you have it. It's the same as your slide. So it reacts with an enzyme that belongs to the oxidase families, and which produces the ketones. These ketones, they will go through a cascade of a chemical reaction to the end produce the melanin, which are ended with the protein, which what we call a complex melanin protein. And this is what, these are melanin proteins. They are the one responsible for the brown color. So to remember from this slide, so you will need a polyphenolic compound that reacts with an enzyme to produce a quinone that goes through a cascade of a reaction to the end gives us melanin or melanin complex protein. This is the one responsible for the browning. Here, we keep seeing the same thing, AW curve. As you can see here, the ability of water, so the enzymatic activity starts at 0.3 and increases to reach its top around 0.8. Very high or higher AW above 0.85 or above 0.9, there is no enzymatic reaction. So, and in this case, because of the water that is available, so it is able to act as a solvent for these reactions. When we talk about fruits, we talk about maturation. Maturation means metabolic activities. So here, more metabolic active a plant is a shorter life. So the shelf life of a plant, of fruit or vegetable is inversely associated with the metabolic activities. So as you can see here, the one that this is a curve, the figure, so it gives you the rate of respiration, and here it gives you, in the X, Y, it gives you the shelf life. And you can see here, higher respiration life, lower shelf life, and etc. And then on the top or on the upper level of this figure, we find fruits, and in the bottom we will find mainly cereals and legumes. Here, because of their respiration, it's also because of the water content. So water content is the main factor that triggers this shelf life of this respiration, etc. So what happens during the ripening or maturation? When we talk about ripening, we talk about maturation, softening or increasing of sweetness, and change of color. So color can change from green to yellow, or etc. It depends. What we have here, the ripening is due to the metabolic activity of fruit before and or after harvest. So it requires plant hormones, and here we'll be talking about ethylene gas, to be able to stop this ripening to increase the shelf life. What we can do, we can silence the ethylene-producing genes. So this is a GMO. So if we silence the ethylene making or producing gene in a fruit and vegetable, we stop this ripening, because this is like a major component. When we talk about ripening, we also talk about respiration that provides energy. So we have a sugar that reacts with the presence of oxygen, etc., that provides energy and heating, an increase of temperature, which later lead or trigger or increase the rate of maturation. We talk about protein synthesis, so treatment of plant cells with specific inhibitors of protein synthesis. This is, we can inhibit the ripening. So we can easily play with the protein or stop the protein synthesis with different compounds. So this will inhibit or stop the ripening, same as for silencing genes. When we talk about respiration, we can distinguish between two types of fruits. The one that we call climacteric and the one that we call non-climacteric. So why climacteric? This is, climacterics have high level of rates in respiration that coincides with the ripening. And here, the climacteric one, most known common examples are like banana. Banana is a very climacteric one. So they have very high level of ripening and modification. And then we talk about potato, tomatoes, etc. The opposite, we have the ones that generally they mature, ripen before the harvest. So they don't contain merely starch to be able to react and to mature. So the ripening or climacteric, they continue their maturation even after harvesting. The non-climacteric, the ripen stops prior to harvesting. So in here, this is just to give you an idea about the difference in respiration between climacteric and non-climacteric. The non-climacteric, they have the rate of respiration doesn't, absorption of, uptaken of oxygen to release gases, etc., is mostly stable. It declines over time, but it's mostly stable. So that shows that there is no reaction, while the climacteric ones, they keep being active to specific time. So here we can see that it goes to the top. Here I'm not giving you a unit because it depends on the fruit, on the climacteric fruit itself. It goes to the top and and then goes down again. So top because of presence of components or that can be easily uptaken to have with this respiration process. As I said, banana is a good example. When we talk about banana ripening, it's changing of color and softening. Why there is a softening? Because there is a breakdown of pectin. So pectin is a sugar that is present in banana. There is an increase of sweetness. So when banana is green, the starch is present to about 14 to 20%, and then this starch gets digested or like get transformed during the ripening to reach about 1%. So if you remember, starch as a molecule has a lower sweetening level, but then when it is hydrolyzed into smaller molecular way, this is where the sugar or the sweetening increases, and this is what happens during the ripening. And then the color changes. We go from green to yellow, and then it starts maturation to change to become brown, even almost black. Here, when we talk about it's degreening due to the breakdown of chlorophyll. And then here, we talk about an enzyme that is called chlorophyllase. So how do we delay this ripening? So there are two main techniques. Either by trapping, so by imprisoning the gas, the ethylene gas, to delay these ripening. And then here, we either use a modified atmosphere, or mainly we use bags. So, or special packaging. These bags, they contain the potassium permanganate. That this chemical, it binds the ethylene. So when there is no ethylene, meaning there is no ripening. So So the ripening is delayed here. Then we have another way of, and the first one is, it is a modified atmospheric, like mainly it's packaging. And this can be done at a smaller level. So we cannot, for example, we cannot have the potassium permanganate in a room or in storage room. This is not allowed. But then if we want to have modify the atmosphere, for example, in a storage room or in big containers, we modify, we play with the atmosphere. And then, for example, here, we can have low levels of oxygen and a high level of CO2, which balance this. We can have lower temperature. The lower temperature delays the enzymatic reaction. And then which inhibits the respiration and then delays the ripening or maturation of these fruits. And this is what, for example, is done with the apples. So this is why, for example, we can see that the apples are present on the counter all the years, while they are collected and specified. So what is done here, it's they are stored, say for potato, they are mainly stored in modified atmosphere to stop the ripening. Potato it's different, so it's not about ripening. So we'll go fast with this, and then we can maybe we'll switch. We will go with take a break. Akash will give his presentation, and we'll do the revision last. Vegetables, we'll talk about. I'm giving you examples on the Brassica family, Solanae, and the alliums. Brassica family, we have, for example, broccoli, cauliflower, cabbage, etc. We'll talk about Brassica family because they have high content of glucosinolates. So these family of vegetables, they are cruciferous vegetables. They contain the isocyanate compounds. So what are these isocyanate compounds? They are strong odorant compounds that are formed from the glucosinolates. So glucosinolate is the main component, and that would interact with a specific enzyme to give us the isothiocyanates. And this one, it has been shown to have antioxidant effect and anti-inflammatory properties. So this is why, for example, broccoli and kale, etc., they're highly recommended for cardiovascular disease, as anti-cancer, etc. So this is due to the isothiocyanates. When this is formed, it's formed when the tissue is damaged. Same, for example, for the enzymatic reaction or Browning reaction. So the enzyme has to be in contact with the substrate to produce this compound, because the glucosinolates within a vegetable, the glucosinolate and the enzyme are in different cellular compartments. So we need to cut the vegetable to be able to put them in contact. When we talk about the isocyanates, these are compounds that are known to induce the expression of the GST. GST, or glutathione S transferases, these are enzymes, metabolic enzymes, phase 2 enzymes that are present in the liver, human liver, for example. They are responsible for the metabolism. Why these cruciferases are important? Because the isocyanate, it mixes, or it triggers, or it induces the expression of GTAs. And the GSTs, they conjugate the mutagenes and the ROS, or the reactive oxygen species. And this is what they lower the mutagenesis and the oxidation of cells. So this is what happens. Then, for people who are taking, is anyone taking nutrigenomic course with Dr. Al-Suhaimi?
The third year, probably what it is. As a third year student, it's probably what it is, yeah, right?
It's not, yeah. Read it, you're here. So you may take it next year. So you may see something like this. So if you're taking 488, like the slide took it from 488 slide. So either way, we'll be talking, you will see it in either classes, you will see this type of example. And then these GST, sorry, this has nothing to do with food chemistry, just for your knowledge. These GST are known to be polymorphic. So you and I may have different GSTs in our body, so we don't react the same way to broccoli and to the cyanide. So there are what we call lower, there are people that have lower activity of GST, and others they have higher activity of GST. I'm not going further in detail, but if you people who are interested, you can, I'm happy to explain what is it. I will give you some explanation in the transcript. This is just for your knowledge. So then we have the anthocyanin. So you remember, it's a pigment, and it has been associated with very good beneficial effect. They are responsible for the color purple, red purple color in the cabbage. They belong to the flavonoid group, flavonoids. So these are, after all, they are polyphenols, pyrophenolic compounds, that they can vary in color with the pH. This purple color changes with the pH. It depends. So, for example, at pH 7, it is a blue. Then at pH 2 to an acidic pH, it becomes red, like very nice red purple color. Then if we change the we increase the pH, it becomes colorless. And then pH 5, it's like a colorless, it changes the color. This is why, for example, people at home, they, when cooking the cabbage, they add vinegar. And I remember my mom was saying, oh, we add vinegar so it doesn't smell. Yeah, so yes, I don't know, but adding the vinegar or lemon, etc., acidity, this is what it brings it to pH around 3, 4, and this is why the reddish or like very nice color of the cabbage is maintained. And sometimes, like they're cooked and it's colorless, so it's not very pleasant to see and very pleasant to eat. So this is due to the pH. Then we have another family, the Sonolaceae, so potato and pepper. Potato, they are tuber vegetables, so they can be white and yellow flashing. For your knowledge, if you don't know, Yukon that we find in the grocery stores, that is a yellow fleshed potato, it was developed at the University of Guelph. This was in the 80s, and it has been commercialized since then. So remember, canola, it was developed in Canada by Canadian University, canola oil. Here we have, and there might be other examples. So when we talk about yellow color in potato, this is due to the carotenoid pigment. So it's due to these two, lutein and the vioxanthines. When the potato are exposed to light, the potato will turn green. Why? Because this is due to the synthesis of chlorophyll. This is not harmful. And then, but the light exposure or the continuous light exposure, it also stimulates the formation of the alpha solenoids. The alpha solenoids, or what we call the glycoalkaloids, these are considered as 10 toxins. So if potato consumed, raw potato consumed before cooking, they can be toxic to a certain extent. And so this is what, so it's kind of a protection against predator. It's like a way of potato to protect against predators, animals, for example, before being collected and cooked, etc. So these glycocolloids, they are responsible for the bitter taste. They are can be heat sensitive to a certain extent. So this is why cooking at higher temperature will inhibit these toxins. And then they are toxic especially in larger concentration. Don't remember, it should be 15 milligram per 100 gram of potato. I don't remember. If you're interested, I can give you the LD50, the doses of the lethal dose. So this, if potato is eaten raw, it can produce a burning, it can lead to a burning sensation in mouth and flu-like symptoms. So in severe cases, because these can be neurotoxic, these are glycolloids, they can be they are neurotoxic. They can have neurological effects. Pepper, we have sweet pepper, so we have peppers because of different colors, different pigments, etc. When we sweet pepper, when we talk about green, that is where we have a high level of chlorophyll. When we talk about yellow and red, we talk about presence of carotenoids. So usually, the pepper can be green, so high level of we'll see the next one, high level of chlorophyll compared to the carotenoids. So these are, as we said, they are polyene compounds. They have, we'll see it in the next figure, they have, can have double bond in different at different position. Here it's a little bit blurry, but they're double bond, and this is what they're responsible for the colors. Carotenoids are characterized, as I said, by conjugated or alternating double bonds. So here, when we talk about green, orange color, it's carotenoid. Green is the zeaxanthin. The yellow is the lutein, etc. So these are what makes the pepper different in color. Hot pepper. Hot pepper is due to the presence of the capsaicin. Capsaicin is, this is what, it's a molecule that can trigger specific receptor in the mouth. And to the best of my knowledge, it is a it's a solid in lipids. Solid in, it's liposoluble, solid in lipids. So this is why, for example, if we want to mitigate the sensation, people say, oh, you drink milk or you eat butter, etc. Because these are liposoluble, so they can easily be solubilized in lipids or oil or whatever. So this is what makes these pepper hot, and they have been associated or shown to have anti-cancerogenic activities. How the color changes? When we have green fruits, we have biosynthesis of chlorophyll. So the chlorophyll is higher level, what makes the pigment green, compared to low level of lutein, violexanthin, and the beta carotene. During the ripening, there is a change of biosynthesis, and here we have a decline of the chlorophyll and the lutein, and we have an increased level of the beta carotene and violexanthin. So this is why the color changes from green to becomes yellow or becomes orange, etc. And then here we can also talk about de novo biosynthesis of reg pigments. And it's mainly due to this biosynthesis of the xanthine. The last slide, allium's family. I know many of you, at least 2 or 3, had worked on allium in your report. So they are a family of garlic, onions, etc. They have characteristic aroma and flavor that is caused by the presence of sulfur compounds. These precursor compounds are catalyzed by an enzyme that is called the allinase when the tissue is damaged. So why the tissue has to be damaged for this? The same as the isocyanate or glucosinolate, same as Browning reaction. So this is because that issue or the tissue damage, here there is a tissue. The tissue damage causes the release of the enzyme and acts on the precursor compound. So we have, because the enzyme and the substrate, same as other vegetable, enzyme and substrate are not in the same compartment, and this is why we have to cut or damage the cell to make them together. When we talk about garlic and onion, they have different flavored compounds that contain sulfur. Garlic, it has the allicin, or the thiosulfonate. And the onion is known for the presence of the thiopropyl S oxidase. What is this? This is what makes us cry when we handle. This is a tear. I don't know how to draw tear. Should have done something. Should have looked at. I should have done a better word. Just imagine this is a tear. So why? This is due to the presence of this compound, and this is what we call the lacrimator. So a lacrimator. Lacrimator. So this is the compound that makes tears. I will be posting self-assessment, same as last time, and then to wrap up, we're not going to wrap up the message. We'll take a break. I know I'm not respecting what we said last time, but we're making exceptions. And then we can give the microphone to Akash, and then the last part we'll do it. Okay? So let's take a break. Any question? I went about 20 minutes overtime. Be recording.
Yeah.
Hopefully it won't. Okay, so back after the break. Okay, so we're starting our guest lecture. So I'm very happy and proud to have Akash as a guest speaker today, who will be talking about something that is, you will see, very interesting. So I'm not going to introduce him, but he will introduce himself, and he will tell you about what he's doing and his research, etc. I'm proud because he's my TA. I'm proud because, so I knew Akash last year?
Yeah, last summer?
Yeah, last summer.
Yeah.
Akash came because when I was just to this.
Sure. Well, thank you for those very kind words. Can everyone hear me okay, or should I go to the mic? Is that fine? Okay, sounds good. All right, well, good morning, everyone. My name is Akash, and as you have been told, I'm one of your TAs for this course. And today I'm delighted to be giving a talk on plant proteins. And we're really going to focus on the real-world impact of food chemistry and food processing in relation to health. So I hope you can come away from this with a greater understanding of how the things you're learning here can be translated into different applications. So just a brief overview about me. I did my undergrad here at U of T. I majored in immunology nutrition, and as Mel said, I did take NFS386 food chemistry, and then I completed 2 thesis projects with Drs. Thomas Eiwiger, who's a clinician scientist at Sick Kids, where we focus in food allergy, and then also with Dr. Deborah O'Connor, who chairs this department, and she leads a rather large research program in human milk. So that's how my master's project kind of got created. So currently I'm finishing up my master's at Sick Kids, or in the Institute of Medical Science, and most of my research takes place at Sick Kids. And typically all our research is in food allergies, and as you can imagine, therapies for food allergies naturally encompass plant proteins and food processing. So just the last little thing about me is for my actual project, in case you're interested. We look at sort of a multiplex sensitization kit. So we look at allergies, or we essentially profile an allergic patient for all different sorts of proteins. So this is actually a sort of microarray. It has 300 different proteins on it, coupled to nanoparticles, and then we can scan it and essentially just incubate with patient plasma or serum or their blood and see what sort of profile they're allergic to and what shows up for things like cross-reactivities and what's a true allergy or what isn't. So, and for me specifically, we're looking at this in breast milk and seeing how maternal sensitization patterns can impact in their offspring and their child sensitization patterns. So that's enough about me. We're just going to have a brief talk or an overview about the different sections for plant proteins. I'm just going to focus on mostly the grasses and other crops, legumes, fruits, and then pollen, since these are the kind of the major four. Yeah?
Can you be just closer to the microphone?
Oh, sure, of course. Yeah. Is this better? Oh, is it on? Oh, sorry. Let me see. Hello? No, that's fine. I can speak louder if you'd like, yeah. Hello? Hello? Oh, wow, okay. There we go. That's a lot better. Let me just move this over here. Sorry for the delay. Great. I hope that's better. Okay, yeah, so I'm just going to talk about grasses, legumes, fruits, and pollen. So you can see the 4 major plant proteins of major interest. So to start off with the grasses, as you know, many of them are cereal crops, so they're cultivated for the grains. Some of the more common ones are wheat, rice, corn, rye, oats, things that you're very familiar with. But the important part is that they're a good source of protein, and the protein that we're going to focus on is gluten. And I'm sure you've already talked about this in your other lectures earlier in the semester, but gluten is processed from the ethanol extraction from things like wheat, barley, and rye. And the key part here in terms of the protein is that, let's say you have a slice of bread, which is about 25 grams. Generally, about 10% of it is gluten. So in a 25-gram slice of bread, that's about 2,500 milligrams, and you'll see why that's important in a second. So for legumes, these are also seeds that are very important, not just for protein, but also dietary fiber. However, several proteins are potentially toxic, but they can be modified or destroyed. So by toxic, I mean they can be allergens. They have things like proteinase inhibitors, alpha amylase inhibitors, lectins, and glycoproteins that can be harmful to the body. And some examples that are well known are soybean, peanuts, lentils, chickpeas, peas, things that you've seen perhaps in your life that people are commonly allergic to. And I don't know if you've already taken earlier biochemistry, but the real dogma of biochemistry is that protein structure dictates function. So when I'm talking about how can they be modified or destroyed, if you can change the structure of these plant proteins, you can probably change the function as well. So this is really important for things like food allergies. So the next section about fruits, they're, as you know, part of a healthy diet to prevent many disorders. And as well, they also have key proteins and allergens, despite being if it's raw or if it's processed for consumption. And this can be anywhere, really. It can be in the outer layer, the peel, or the pulp, or even the seeds. And the important thing about fruits to know is that cross-reactivity between fruits, if you're allergic, is actually quite high, since there's such evolutionarily conserved structures within these proteins. And you'll see how this ties into the pollen. So pollens are produced by many plants, especially during the spring, summer, fall, and especially when certain trees and grasses pollinate. And they often contribute to seasonal allergies, things like hay fever, allergic rhinitis. And typically some are perennial, but usually they fluctuate between different seasons. For example, trees tend to be more active during the earlier months in April to June, things like birch, oak, elm. Grasses in the summer, things like Bermuda grass, Timothy grasses, or really any other type of grasses. And the molds tend to be later in the year, things like ragweed, the cladosporum, the alternaria alternata. And these are also highly cross-reactive proteins with fruits and vegetables. As you know, fruits are essentially ovaries of plants, so you can imagine they're highly cross-reactive, which now we'll move on from the overview and explain what I mean by cross-reactive and get into the protein interactions. So specifically for gluten, since we're talking about the grasses, gluten, as you already know, I've learned, is made up of gliadin and glutenin. And the gliadin is the more important one in terms of your immune system because it can stimulate it through the different peptides linked in as part of that structure. So particularly it has a lot of proline and glutamine sequences, and these are very stable and helps prevent complete digestion by the intestinal enzymes. And this resistant fragment of gliadin, this 33 polymer peptide, is what's known to cause celiac disease symptoms. So in terms of celiac disease, it's an inflammatory digestive condition from an immune reaction to gluten, as you might have already known. And this damages the ability to absorb nutrients from the small intestine. And we were talking about the slice of bread beforehand, how a 25 gram slice of bread has about 2,500 milligrams of gluten. And for specifically to celiac disease, it's not exactly an exact science, but typically the daily lowest amount to cause damage to your intestinal mucosa over time is about 10 to 50 milligrams. So it's not a lot. This is only about 1% of a slice of bread, so essentially a crumb. And in terms of actually labeling for food processing and chemistry, in terms of labeling on actual foods consumed, 20 parts per million is the FDA limit for defining something as gluten-free. So it's allowed to have about 0.5 milligrams in about a slice of bread. So again, that isn't a lot as well. So for legumes, peanut and soybean account for a significant portion of food allergies in Canada. They're among the priority allergens that most prepackaged foods are required to carry a label for, and this is set by Health Canada and the Canadian Food Inspection Agency. And by priority allergens, I mean they're considered to be among the top 10. So the top 10 being things like peanuts, soybean, egg, wheat, fish, shellfish, sulfites, etc. And those account for 90% of food allergies in Canada. And just to give a little background, since I don't know how familiar we are with the immunology, an allergen is something that's defined as a usually harmless substance to which your immune system responds to, and this is a physiological overreaction. And these food allergens can even be found in non-food items such as medications, vaccines, cosmetics, children's crafts, or even in the body fluid to others who have recently ingested these food allergens, such as saliva. So just to give a bit more detail into allergies, an allergen is also known as an antigen, and if you want to go by the immunology definition, antigen being antibody generating. And these are usually proteins, but sometimes glycoproteins, as you'll learn later on in this lecture. But the important part is that they're usually resistant to heat, acid, digestion, but it depends on the component proteins and families that the protein, or specifically plant protein, that it's a part of. And plant proteins often fall under the prolamin superfamily. I think you guys have talked about this briefly when you're talking about wheat and gluten. But there's other families that this can be a part of, such as profilins or tropomycins, which are the major proteins in seafood and seafood allergies. So just to recap what a prolamin superfamily means, it means that these plant proteins have conserved evolutionary structures that are common throughout different foods. So for example, we have these non-specific lipid transfer proteins that you can find them in different fruits and peaches, apples, pears, nuts, different legumes, cereal grains, and they all have different functions and categories. But generally, for example, the 2S albumins, which is a type of protein, tend to be very resistant to heat and degradation. So what happens when you actually have an allergic reaction? So upon first exposure, you're actually technically not allergic yet. It's called sensitization. Your body creates IgE antibodies against this protein, which it shouldn't be doing. It's a bad case. And so essentially you're having antibodies when you shouldn't have them. So when you get re-exposed to them, you have an actual reaction. So what happens is that antibody that's generated binds the protein, binds the allergen, and the recognition via the cell surface receptors triggers an immune cascade. And I won't get into the details of the immunology since this is not an immunology course. But essentially what happens is that you have a local inflammatory response, or if you have really severe allergies, you can maybe end up with systemic anaphylaxis. So the analogy I like to use is, imagine your neighbor is coming over to say hello, but you don't know why they're there. You freak out, you don't know why they're there, and you start throwing possessions and furniture at them because you're trying to get them out of your place, but in doing so, you completely crash your home to the point of it being unlivable. So that's essentially what happens with an immune reaction, which I'll show in the next slide. But just a side note, since we're talking about IgE antibodies, there's actually quite a few different categories, but for the purpose of this class, we're just focusing on true food allergies, which are a type 1 hypersensitivity reaction. So these are some of the non-IgE antibody-mediated hypersensitivities. I'm only mentioning them because they're also food or plant protein-based typically. Things like FPIES, food protein-induced enterocolitis syndrome, food protein-induced allergic proctocolitis, or even food protein-induced enteropathy. But these are typically non-IgE mediated and they're very delayed. So instead of minutes to seconds, it's hours to days for this delayed reaction. They're typically not as life-threatening, and they're typically also usually found only really in infants and resolve in a few years. But the management is still the same for food allergies as well as this. It's generally avoidance, and the physiology is very poorly understood. Specifically for these, it's also very similar. The triggers are usually milk, eggs, some grains. But they have different therapies as well as available for it. Anyhow, so back to legumes. So plant proteins can be responsible for major health issues, which is why we're talking about it, in a significant population. So celiac disease has a prevalence of about 1% in North America, but about 10% of children in North America have a food allergy, which some of them outgrow, some of them don't. So given this high incidence and associated healthcare burden if children are being hospitalized for this, are there non-pharmacological interventions available so you don't need to give epinephrine or adrenaline to treat these bad reactions to plant proteins? So clearly the problem is the food or consumption of it. Can we modify these foods to be either less allergenic, or can we actually modify them to be a treatment, similar to how you can imagine a vaccine where you're given a small little dose or small portion of the actual agent, and then you create antibodies against it so you can fight the real thing. So what happens essentially with this allergy is your body wants to fight this perceived created problem so badly that it opens up your blood vessels as much as it can to get your white blood cells, your immune cells, there quickly. But that's a problem if you do it everywhere in your body. If all of your blood vessels open up simultaneously and cause hives, swelling, your throat closure, you can really die from the asphyxiation or the very, very low blood pressure that is associated with this food allergy. So in terms of fruits and their protein interaction, typically we're talking about the Rosaceae, the family. That's the most common trigger among fruit allergens. There's different types. There's perennial things like apples and pears. There's stone fruits, peaches, other things, and shell fruits like almonds. And we talked about a bit of prolammin family and other protein families. I've highlighted it because it's important that things like pathogenesis-related protein families, that when we're talking about food modification, some of these protein families are much more resistant to degradation. So the PR10 protein family is actually expressed in most plant tissues, and it's typically upregulated upon stress or attack of the plant. But these proteins are very liable at extreme pH, heat treatment, and even endogenous inhibitors like polyphenols. But there's also other protein families within these fruits and other vegetables, such as the nonspecific lipid transfer proteins. These are much more resistant to processing, and they're often typically found in the outer layer of these fruits, such as the peel. So if you've ever seen fuzz on an actual peach or other types of fruits, that's what it actually is, the protein. So pollen is where it gets a bit interesting. So pollen, I've mentioned, has a lot of highly cross-reactive components between their proteins and with fruits and vegetables. So BETV1 is called a major birch pollen allergen, and it really can trigger a reaction from other different fruits and vegetables like apples. I don't know if you can see this very well, bell peppers, things like that. And the good thing about this is that if you avoid eating the raw or fresh version, if we can process these foods, typically they can be well tolerated, which is great. And there's so many complex interactions that are still being discovered. For example, if you have a latex allergy, typically you can't, you're sensitive to bananas, kiwis, other different fruits as well. So these pollen cross-reactivity is a really big thing, and it's called pollen food, well, not food allergy, but food sensitivity syndrome, or used to be called oral allergy syndrome. But the key point here is that if it's processed, if it's baked, if it's in a canned form, if it's in a peeled form, people can sometimes tolerate something like apple pie, but they can't eat raw apples due to this cross-reactivity between plant proteins. So that's it for the protein interactions. Let's talk about the actual protein modifications in a sense. So as we discussed, protein structure dictates function. So how can we change the structure? How can we process the food to be beneficial? And which goal are we trying to go for? Are we trying to mitigate these adverse health outcomes by reducing their chance of reaction? Or can we actually try to attempt it as a treatment using food chemistry? And the main considerations for talking about protein modifications are the enzyme activity, the stability, how resistant is it to pH or heat, and its glycosylation. So let's talk about grasses. So there's actually been quite a bit of study in terms of wheat specifically. I'm only going to cover a few of the different methodologies. One of the big ones is thermal processing, just for examples. For example, they found that unheated bread dough versus the baked version of bread, when you process it thermally, these induced protein aggregates that form prevented the proteolytic breakdown. So what that essentially means is that when you create that sort of glutinous, nicely delicious baked bread, the entire aggregate of this makes it more resistant to being broken down by your enzymes, and essentially this is not beneficial in some cases because it allows the passage of this through your intestines, and it can bind to these antibodies and trigger them. But it's different for each food and even different food process. So something like pasta, which is usually durum wheat, to create the pasta, it undergoes extrusion and drying, but also at different temperatures. And it's also typically boiled before consumption. And all these things can affect protein binding activity. So I'm not going to go into details of the papers or specific mechanisms, but I just want to tell you about different processes that can affect protein binding affinity and affect allergenicity in a sense. A few of the more interesting ones are fermentation and hydrolysis to modify proteins. So acid hydrolyzed wheat gluten is actually used as an emulsifier in foods and cosmetics such as soaps and shampoos. And because it's acid hydrolyzed, they can create new epitopes, so protein binding sites to an antibody, and it can also alter the sphericity. Things like hydrolysis with proteolytic enzymes are actually often used to create hypoallergenic formulas and flours, since you're essentially changing the protein structure. The more interesting one I actually came across recently is 2 studies evaluating the potential of some specific lactic acid bacteria and even a mixture of probiotic lactic acid bacteria and bifidobacteria in the production of sourdough to hydrolyze these wheat flour allergens to see if people have a less reaction, either if they're celiac or if they have a true wheat allergy. So legumes, in terms of thermal processing, here is actually where it gets a bit confusing in terms of the literature. It can either go really both ways. So tree nuts are actually quite often heat processed, either dry or moist. You've probably seen now plant-based proteins, cashew butters, almond butters, everything else like that. But it really depends for thermal processing. They've tried blanching, they've tried roasting. For some nuts, it's shown beneficial effects like hazelnuts. For peanuts, roasting them is actually really shown to increase their allergenicity for whatever reason. Typically, the thought process behind that is the Maillard reactions. So you've seen this glycation of reducing sugars and amino groups before in your lecture, and it's thought that the roasting of the peanuts may increase advanced glycation end products, which may contribute to this allergenicity. But it's really unclear, the literature for what these thermal processing can do to legumes, since they're so resistant. The seed storage proteins in them are so resistant to digestion and manipulation that it's really not sure yet. So we're actually really trying a lot of different avenues for this, things like autoclaving, microwaving, even frying or steaming, and even boiling as well. So in terms of the actual mechanism of the biochemistry behind it, boiling of peanuts has been shown to reduce the binding capacity of IgE antibodies to the protein, so tend to be less allergenic. And the reason it does this is it actually changes the alpha helix and beta strand structures in post-processing of these proteins. And as you can recall, many allergen protein families exist, so the prolamins, the profilins, the tropomycins. Many are resistant to degradation, but as long as the protein is still intact in a sense, it can technically cause a reaction. And usually allergens are protein-based since they bind to receptors, which is mostly, as you know, enzyme and protein-based. So in the theoretical sense, highly refined peanut or soybean oil is not really allergic, but cross-contamination is an issue. It's pretty hard to get rid of every single protein within the nut. And as well, there's some carbohydrates that can be glycoproteins and bind to different structures. So in a sense, people who aren't really severely allergic to peanuts or soybean can consume the oil, or even things like sesame oil, but it must contain no protein. It has to mostly be pure lipids, the pure lipid fraction. So this is where processing issues can come into place. So one of the legumes I want to focus on is soybean because it's typically the one that's been mostly processed. As you can see, there's a lot of different ways to convert soybean to different things: soy sauce, miso, tempeh, lecithin as an emulsifier, gums, oil, TVP and tofu, and things like that. Typically has the most food processed, and I wouldn't say everyone's allergic to all these different components, but there's a lot of different ways to manipulate soybean and novel ways as well. So things like using pH to change the binding affinity of the protein, things like high pressure, hydrostatic pressure processing, which can break the non-covalent bonds under specific conditions, other techniques like using pulsed ultraviolet light, or even using pressure to change the atmospheric pressure around the protein in a controlled environment to see if that changes the protein structure. So for fruit and pollens, I've mentioned that, again, they're highly cross-reactive, but usually the raw version is the one that's the issue, not the processed version. So peeling, many fruits can be tolerated without the peel, and the fetus had the pulp. And there's a proposed, and you can do this mechanically or chemically peeling for some fruits. But there's other techniques since most processed forms are well tolerated. There's not much literature around it since it's so well tolerated. If it's not an issue, generally people don't like to do science about it. But yes, baked versions, canned versions are usually okay. But in terms of cross-reactivity, it's a big issue. For example, quite a few, as you can see the number, 30 to 70% of people who have actual true latex allergies can also react to bananas, avocados, kiwi fruit due to the cross-reactivity. So modifications for these proteins are kind of important. So that's it for protein modifications. Now we're into the really cool stuff. How can we use this as therapy? So we're going to talk about immunotherapies, where this has gone wrong. How can we substitute these plant proteins for things that are less allergenic? And a really cool, interesting case report that I want to discuss. So this is a bit more immunology for you, but it's a very good treatment option for some allergies. It's typically very commonly used for pollen, venom, some select foods in a very controlled environment with very stringent inclusion criteria in a select population, and it's called oral immunotherapy. So what this means, it involves continuous ingestion of, let's say, the offending allergen in a controlled setting, and typically there's a stepwise increase in the different doses. So say you're mildly allergic to peanut and you want to increase your threshold for a reaction, you can essentially microdose at very small quantities in milligrams or micrograms in a controlled environment in a doctor's office in the hopes that you can raise your threshold so you can consume something like a may-contain product without worrying about a severe reaction like anaphylaxis. So the goal is essentially to consume the allergen or at least consume the may-contain amounts. And as an example, a single peanut has about 200 milligrams of protein. If you talk about gluten, we were talking earlier about, let's say, 10 milligrams is a threshold for sensitivity. The only caveat of oral immunotherapy is typically it's not invasive, but it takes a while. So you have to do it for quite a few years. And to maintain your desensitization and your ability to eat this without having a reaction, you have to continue eating it lifelong. So usually at least 2-3 times a week for the rest of your life if you do undergo this therapy, and it's not for everyone. There's quite stringent criteria. And of course, there's a risk of reaction during therapy since you're trying to essentially figure out what is my threshold, what can I eat at a safe level. But it is very effective, especially in young children. And I'll explain what I mean. So specifically for milk, very young kids and like preschool children with mild milk allergies, which in general, cow's milk allergy is typically outgrown. About 90% of them outgrow it. But for those who don't, Canada's actually come out with a guideline for anaphylaxis called a milk ladder, essentially, but they've done this for egg as well. Since most can tolerate the extensively heated or modified form of this protein, they've created a ladder for like a stepwise based approach you can do at home, so you don't need to go to a doctor's office. And typically you start with the baked products, something like a cupcake or a cookie, and then you make your way up. If you tolerate it for a few months, You can make your way up after getting approval to the pancake. Then you gradually increase the amount of protein or cheese or dairy component that has the casein, that's typically the trigger food. And you eventually make your way all the way up to milk or cheese. So this has also been done for egg. And again, typically the same slides, but the baked version, muffin cupcakes, egg noodles, French toast, and then you're into the actual eggs. Because these allergies are commonly outgrown. Peanut, it's a lot harder to do this for, but they are trying to do it for not just single allergens, but they're doing multi-dose oral immunotherapy, multi-allergens in a very regulated environment. So that's for food immunotherapies. For pollen, it's actually very well established. There's numerous options if you have really bad seasonal allergies to pollen. You can do this subcutaneously, sublingual, under the tongue. It can be a tablet, it can be an injection, but you do have to take it often to get protection from these plant proteins. For example, you have to do it for 3 years, and generally there's actually some pretty good long-lasting studies that show if you do this therapy for 3 years, you get about 7 years of protection, and then it kind of rebounds back up if you stop it. So it is good, but it does take a lot of your time to have this therapy for 3 or 4 years, and then you get this nice effect, and then you have to repeat it. But it is good to avoid these pollen allergies. The other option is also wearing a mask, which typically reduces the amount of pollen in your face. As you know, people in Japan have been doing this for probably the better part of half a decade. But masks, of course, protect not just against pollen, but other things that can get in your face and cause irritation. And specifically to pollen, climate change has actually really increased the season and the length of the season. Normally, I said we started in about March or April, but pollen counts were actually a historic high last year, and it typically been actually in February. So it's a very exciting field that people are looking into for seasonal allergies. So that's good therapies. Here's the bad part for protein reactions. So I'm personally vegetarian, so it doesn't really impact me as much. But you may have seen in the news a thing called alpha-gal syndrome, and it's attributed likely to the Lone Star tick bite in the States, so think southern U.S. states. And the reason behind this, I think Emil actually talked about this earlier in the class today about alpha-gal, but essentially this is a sugar that's not produced in humans or primates, typically in other animals. And this tick essentially can inject alpha-gal when it bites you, and you produce IgE antibodies against this. So as a result, if you're having red meat products, you can get a delayed allergic reaction because of these antibodies that are reacting to the sugar in these meat products. So food processing hasn't been shown to help yet. So the solution right now is avoidance, or you can consume plant-based protein, or consume a meat from that doesn't contain alpha-gal, such as poultry, seafood, things from reptiles, etc. In terms of plant-based protein, one of the more common ones you've probably seen for sure is Beyond Meat. It's typically in most of the food chains now, like Tim Hortons, etc. And the main protein source that they use is pea protein. And some people are allergic to this, so it's not for them, but it is a good option if you're trying to avoid meat and reduce your consumption. It contains pea. The main ingredients are here: pea, mung bean, fava beans, all these healthy legumes with a lot of protein. They also have fats, and the goal is trying to recreate meat essentially. So they have other ingredients to how to get the flavors: calcium, salt, iron, potassium chloride, things that help bind the burger itself, potato starch, methylcellulose, and then things that mimic the appearance and flavor as well. So beet juice that gives it its redness. There's other extracts and natural flavorings in it. But essentially, plant-based proteins can provide different options, and it is worth exploring since we're talking about this course about plant proteins and how they can be used. So they can create essentially fake meat. But there are differences. For example, here on the right is Beyond Meat, the nutrition facts for it, and to the right of that is ground beef, which is 90% lean and 10% fat. So if we actually compare them, there's significantly greater amount of calories in Beyond Meat, also significantly about double the fat and about 4 times the sodium. So it's not exactly as healthy as everyone is proclaiming it to be. It's an option as a plant-based protein, but you still have to be careful and watch where you're eating. Just because it's plant-based doesn't mean it's exactly healthy. If you're actually interested in looking at other examples, you can look up lab-grown meat. So if you think of a typical lab, you can actually grow meat fibrils from myosins. You talk about your meat lecture, but they are creating lab-grown meat purely from essentially cell culture, which is also really cool, but that's a bit farther away. But yeah, the overall point is that I'm trying to show you is the applications of plant proteins and how they can be used. So here's the case report. This is what I was really interested in talking about, published this summer. It was a clinical case report published in JACI, one of the immunology journals, and it described anaphylaxis from mango puree, which was contaminated by mango seeds. So there was 2 young children who had peanut and cashew allergies. So young children being 2, so they're still having baby food essentially. They previously had mango before, mango puree from another manufacturer. They've had the fruit pulp, etc. But then they reacted to this specific manufacturer's brand of mango puree. So they did allergy testing, skin prick testing, and serum testing, which I won't get into, but they tested the mango puree, the original baby food, the fruit pulp, the mango peel, and the mango seed, which is mechanically ground. So it turns out both the children were actually allergic to the seeds. So if you look at a mango on the right, there's the endocarp, the mesocarp, and the exocarp. I don't know if all of you have tried a mango before, but typically you don't consume the seed. It's not for consumption. It's sliced or eaten around. But essentially both these children were fine with the pulp and the peel, but they are allergic to the seed. And the reason the seed became an issue is because it's so evolutionarily conserved as a seed storage protein. There's a lot of cross-reactivity with cashew and pistachios. So why would this happen? Essentially, if you don't usually eat the seed, you would never come across this. But because the baby food company revealed that they essentially had to investigate this, but the baby food company revealed that the whole fruit, sometimes in the process of making this mango baby puree, it's sometimes crushed and minced, so it includes the seed. So essentially the manufacturer could not rule out the possibility of traces of the seed, and as such, they had no obligation to change the labeling on the actual packaging, because mango is not a nut in itself. There's no nuts in the puree, so it's not deemed a priority allergen. They don't have to include it as their ingredients. Typically, most directives require labeled ingredients for foods derived from nuts, but not things that might be cross-reactive with nuts, such as a seed storage protein. So really an upcoming field is how do we detail these food processing methods, and how can we change the guidelines so people are not ingesting things that are cross-reactive that may trigger their allergies. So since this ingredient, the mango seed, is not intended for consumption, it's not a natural part of the produce, since typically you just eat the peel or the pulp, how can we change this? Should we be able to put on the actual packaging may contain unexpected ingredients, or may contain the seeds or kernel of the actual plant or the protein? Or do we just say things, or more blanket statement, not suitable for consumers with other allergies? So it depends how you really want to get into it. But I thought this was a really cool case report, and directly shows how food processing can be good and bad in some cases. So yeah, that's everything from me. Thank you. And if you guys have any questions, please feel free to ask literally anything, course related or not course related. But yeah, that's it. All right. Mel, do you have any questions? If not, you're more than welcome to email me, whether if it's course related for TAing or just the lecture itself. But I thought it would be a really good overview of the impact of plant proteins, rather than just the food chemistry, but how can it impact stuff in real life. All right.
Changes from chemical reactions.
Sorry?
Changes from
Yes, yes. No more organic chemistry molecules you're looking at, yeah. Mm-hmm. All right. Great. Okay, then I'll just unplug.
Thank you very much,
Yeah.
Akash. This was very interesting.
Thank you.
Thanks for sharing what you're doing.
Mhm.
So let's continue. Just take 3 minutes break, and then we can continue with the revision.
Yeah, sounds good.
Anyway, if you have any questions for Akash, you have his email. You can post your question on the discussion board. Right?
That's pretty good. 45 minutes. It only goes too long. Probably like 40 minutes or something.
It's always like this.
Yeah. I don't want to talk for an hour. Did you learn anything new? Did you learn anything new? Or did you read all this stuff? I think I told you most of it, but yeah.
Yeah, no, I learned something new.
That's always good.
Yeah. Yeah, no, I like it.
I liked your like, you know, linking the meat and like vegetable lab meat.
Yeah.
You know, there's something.
Yeah, that meat's getting pretty
People need.
unpopular actually. A
Yes.
lot of people don't want to have it.
Yeah. This weird.